No, you're messing up our shot.
We're gonna get stormed on.
That'd be cool if we're like giving this a rating.
Welcome back to the Minnesota Lake Lookout Project. My name is Hilarie Sorensen. I'm the Water Resources Extension Educator with Minnesota Sea Grant.
Hi, I'm Chris Filstrup. I am a limnologist with the University of Minnesota's Natural Resources Research Institute.
And today we are going to be filming our second video tutorial for this project. Way back in the beginning of 2026, you saw us out on a lake that was frozen at the time. We drilled a hole in the ice and talked about collecting water samples for looking at chloride concentrations. Today you can see we're in a little bit of a different environment, and today we are going to be focused on collecting a water sample if you see an algal bloom on your lake. All right, so just a quick reminder, Minnesota Sea Grant and the Natural Resources Research Institute, in partnership with local organizations, lake associations, soil and water conservation districts, we are working to build a community-based monitoring network targeting two pressing water quality problems. One is chloride pollution, the other one is Cyanobacteria harmful algal blooms. We call them Cyanobacteria blooms, harmful algal blooms, or HABs for short. So as we refer to these throughout the video, we're going to talk about HABs. Monitoring these two water quality problems is difficult because they are both episodic and unpredictable. We don't know where and when we might have an influx of chloride into a body of water, or when or where we might see an algal bloom pop up. So today we're going to talk about how to observe these, how to identify them, and how to collect a water sample.
Perfect. Through the Minnesota Lake Lookout Project, volunteers like you can actually help us track where harmful algal blooms are occurring. Because these things tend to form very rapidly and disappear just as quickly, it's very hard at a state level to know where harmful algal blooms are occurring, which makes it really a management challenge, and actually trying to have the necessary information to warn the public about where harmful algal blooms are occurring. So through this project and your great volunteer efforts, we will be able to have more information that we can provide to state agencies so they can better inform people where harmful algal blooms are occurring.
Okay, so by the end of this video, you will know how to safely collect a surface water sample. You'll be able to identify if it is an algal bloom, a cyanobacteria bloom. You'll be able to collect environmental data using bloom images. You'll have a pocket microscope in your sampling kit to do this, and you'll be able to enter the data into a database. That'll be kind of our next step, is how to actually deal with the data once you've collected it. But first we're going to talk about some safety guidelines for when you're out in the field.
So on this project, your safety is our #1 responsibility. Not all harmful algal blooms produce toxins, but some do, and so we want to prevent any kind of exposure to that, especially if you're working in really dense harmful algal blooms, water that looks like pea soup, or also these really thick surface scum that looks like someone poured a layer of paint on the surface of the lake. So if you are sampling, please put on your protective gloves before handling the water and collecting that water sample. I should say, if you do accidentally get some of the water on your skin, it's best just to wash your hands with soap and water to remove any potential toxins that may be on your skin. And you especially want to do this if you're out on the lake and you're going to consume a lot of snacks while you're out there. Definitely wash your hands before eating.
And then aside from just the specifics for how to handle it if you see a harmful algal bloom, the other just golden rules of being safe in the field is always use the buddy system. Don't go out sampling alone. Wear a personal flotation device or a PFD if you're on your boat, or at least have them on your boat. Be careful if you're like we are, kind of in a, maybe if there's some rocky footing or maybe some unsafe footing, just be careful of where you're stepping. And if you do start to feel lightheaded or ill while you're near a bloom, move away from that area as quickly as you can.
Hilarie, when we're sampling today, will you be my buddy?
I will.
Excellent.
All right.
Let's sample.
Let's sample. So, when you are volunteering with this project, you will receive a sampling kit. It is going to be in a cooler like this. You will also receive a clipboard with a laminated water sampling protocol and a few data sheets printed on Write in the Rain paper. Also, you will become registered with an account on the project website, mnsg-mn-lake-lookout.org, and there you will also have digital copies of all the sampling protocols and data sheets, as well as all of the training modules. So this is your kit. Inside you will find 6 of these 250 milliliter amber bottles. The reason we use these amber plastic bottles is to keep sunlight out, to slow down biological productivity once the water sample has been collected. So this is what you will use to actually collect your sample of water. You will also find a pen style water quality meter. This is what you can use to both look at surface chloride levels and then also to take a surface water temperature reading, and you'll record that in Fahrenheit. You will also find some pairs of the gloves that Chris mentioned. So if you do observe a bloom, make sure that you put these on before you start to collect your water sample. And then other items in your kit will be this digital microscope, this digital pocket microscope. We're gonna walk through exactly how to use this, but this is how you can take detailed pictures of your bloom. We also have these nifty project bandanas, which we will show in more detail later, but these are really nice if you're in the field, especially if it's on a hot day, hot sunny day. And then we also have a Petri dish, which you will use for your pocket scope to take a look at the sample, and then your data sheets. And we have a marker for labeling your sample bottle and a pencil for recording your data on the data sheet.
I'm going to go through the data sheet and the kind of information that you're going to enter here. So first, entered by data collector name, I would simply write in my name, so Chris Filstrup. I would write in the sample date if I knew what the date was. It is 6/16 of 2026. And it is about 12:51 p.m., not a.m., because I'm not a night owl. So the lake name, right now we are at the Duluth Harbor. So I would write that in, Duluth Harbor. And location of description. Right here, I would say that I am at the beach near the UMD boathouse. So we're just going to collect a shoreline sample from the beach at UMD boathouse. I would take out my cell phone right now and just open Google to figure out what my lat and long are, and I would record those there. And there's very nice descriptions of what valid numbers are for your latitude and longitude, because right now this project is only within the state of Minnesota, so our latitude will be between 43.5 and almost 50 degrees north. And we will have a negative longitude number because we are in the Western Hemisphere.
Next, we're gonna look at environmental conditions. So what is it like out on the day and the time that you're sampling? So air temperature is gonna be the first thing we'll wanna record. You can just open your favorite weather app to get that temperature in degrees Fahrenheit. We also would like to know what the cloud cover is like. So is it clear and sunny like it is out today? If there's less than about 10% cloud cover, we'd say it's clear. Partly cloudy is gonna be that 10 to 50% cloud cover. Mostly mostly cloudy, 50 to 90% cloud cover, and then overcast or raining is gonna be above 90% clouds. Wave conditions is another important thing to note. So is it calm? Are there small waves? Is it choppy, or are you actually seeing some white caps out there? And then current precipitation is another thing to note, whether it's none or light all the way up to heavy. We also want to know about recent precipitation. So has there been rainfall in the last 24 hours? Especially we want to know if there's been a heavy rainfall event recently, because this is something that can wash nutrients like phosphorus and nitrogen into a body of water, and these nutrients can contribute to the formation of a harmful algal bloom.
And it just so happens yesterday we had a very nice storm system move through Duluth. So here I would probably write heavy storm event, at least where I live, it was pretty heavy.
It was heavy, yeah, so I would note that.
Okay, and we do have a final category that says notes. What should people be entering in the notes section?
Yeah, so if you see any debris, if you see some dead fish floating at the surface, if you see some surface scum or something that's just a little bit different or noteworthy in your sampling spot, it would be great to write that down.
Yeah, basically we want a good idea of why you collected the sample, where you collected it, and to have a little bit of context behind that.
Exactly. The next step is going to be collecting some photos of your sampling site, and you will upload these later when you enter your lake data into the project website. So you can use your cell phone, or if you have another device like an iPad with you in the field, either of these will work. We would like 2 general site photos. So one would be a wide shot looking out at your lake, whether you're on a dock or a boat or a public boat launch, doesn't matter. So you would take a wide shot, and then we would like a close-up tight shot of exactly where you plan to collect your water sample for. So this would be, if you see a bloom, we would love a close-up shot of that bloom exactly where you plan to collect from.
Yep, and the good news about the close-up shot is sometimes we can identify the type of cyanobacteria bloom it is simply by looking at that photo. And this is something we do all the time. People send us photos and say, is this something I should worry about? So there are distinct types of cyanobacteria that you can identify just from an iPhone photo.
Exactly. So the first time that you go out to collect a sample for this project, one thing that we would like to do is to get a better understanding of what your perceptions are of your lake. We do that using this tool called a water quality ladder. This is included, you'll see a picture of it in your sampling protocol and on your data sheet. And basically it's just a ranking from 0 to 10, with 10 being very clear, clean water, with around 8 to 7, 7 to 8 being you think it's safe for swimming most of the time. 5 to 6 is gonna be, yeah, some fish are present. Maybe there are some water quality issues in your lake. 3 to 4 is going to be getting more to that poor water quality side of things. So maybe a 3 to 4 is getting into more limited recreation. There may be some visible concerns, maybe some debris, starting to think maybe that water quality isn't the best. And then 0 to 2 is going to be worst possible water quality. So very poor water quality, has obvious pollution. Maybe there's a fish kill, maybe you see some debris or some trash floating, things like that. So we'd love you to note that just the first time that you sample, and then toward the end of the sampling season to see if anything's changed.
So you can often tell an algal bloom simply by looking at it. So algal blooms commonly make the water look like your grandma's favorite pea soup, or they can form these really thick surface scums that look like someone spilled a can of green paint on the surface of the water. The color of blooms can be anything from bright green to this really interesting like blue-green or cyan color. And that's where we get the common name for cyanobacteria, is actually blue-green algae, because of this pigmentation. But beware, there are often things that we confuse with algal blooms, and I often get lots of phone calls during certain times of year where people are seeing these imposters. One of the imposters is you can look at these long, thick filaments of green algae. Green algae are not cyanobacteria. They don't produce toxins, but they form these big mats that look almost hairy or filamentous when you look at them. Another thing that can be confused with harmful algal blooms are duckweed or pondweed. I think most people have seen these tiny little plants floating on the surface of water bodies. Again, those cannot produce toxins and aren't harmful. This time of year, we are in early summertime up in Duluth, Minnesota, and it is pollen season. I know that because my allergies are going crazy. Lots of times people confuse pollen floating on the surface of the lake as being cyanobacteria blooms, because cyanobacteria can also float on the surface of lakes. One way you can distinguish those is pollen tends to be more this brilliant yellow color rather than a green or a blue-green color. And if you're on the lake and you see pollen on the lake and you're not quite sure what it is, if you go to your car and you have a coating of pollen on your car, it's pretty safe to bet that you're likely seeing a pollen floating on the surface of the lake.
Okay, so now we are going to show you how to actually collect your water sample.
Wait! Don't forget to put on your gloves.
Right. I do not want to come in contact with a bloom, so I am going to put on my gloves. Thank you for the reminder.
All right, so before we sample, we're going to triple rinse the bottle with lake water. And we do this because we just want to remove any debris that may be in that bottle. And so how we're going to do this, you want to be careful not to put your gloves below the surface of the water, because then they're just going to fill up with water and not protect you. So how we're going to do this is we're just going to dip the opening of the bottle right below the surface. And as we bring that up, we're going to swish it around a little bit, which Hilarie forgot to do, but she'll do this time. We'll swish it around a little bit and then dump the water. So when you triple rinse the bottle, you'll want to do this away from where you're going to be collecting your actual sample, because we don't want to disturb that sampling site. So if you're on a boat, you can rinse off the port side and collect the sample off the starboard side. And so after you triple rinse it, we should be ready to collect a surface skim sample.
Here's our actual bloom where we're going to collect our sample from. And what we're going to do is we're going to only fill the sample bottle up about halfway. And this is going to account for expansion when the sample is frozen. And so you're just going to take we want to get the surface of the bloom, because that's where it's most likely that we would find cyanotoxins. So we're just going to do a quick skim at the right at the surface, fill that about halfway. Your sample bottles will have a mark at the halfway point, so you'll know about how much. If you get too much in there, you can just dump that extra out at the top. Put your cap on, and then the next step will be to use your marker, also provided in your kit, to label your sample bottle. So once you've collected your water sample, you will want to label the bottle, and we've provided a Sharpie in each sampling kit. So all you need to put on the label is your name, the name of your lake, and the date the sample was taken, just so we can match it with the data sheet later.
I'm Kimberly.
I'm Allie, and we are both interns working on the mn-lake-lookout project. We're going to show you how to use the digital microscope with your phone or other digital device.
Before you want to head out in the field, we recommend downloading the MaxSee app, M-A-X-S-E-E, from the Google Play Store or the Apple App Store.
Once you're in the field and you're ready to use your microscope, you can connect the microscope to the base using this groove here. And then you just hold the on button to turn on the microscope. There'll be a little plastic cover on the top that you can remove. amazing. So then you could just put your microscope right above your sample, and you can use this slider on the side to adjust the intensity of the LED, and you can use this slider on the side to focus the microscope. And there are also buttons on the back to zoom in and out. Now you can open your mobile device's Wi-Fi and connect to the network of the microscope. It'll be called mnsg, and then a few letters and numbers.
And once you've connected to the Wi-Fi, your app will show a display of the microscope's viewpoint, and you'll be able to take images or even record video.
Once you connect to the scope's network, you can open the app. It'll now show the view of the microscope. You can move it around and focus as needed. You can then hit the blue button at the bottom to take a photo or use your device to take a screenshot.
So to wrap things up on your sampling day, you want to write down any extra observations under additional field notes on your data sheet. Things like, were there any unusual odors at the sampling location or any dead fish present? Finally, you want to lay your data sheet flat and take a clear photo of it that we can back up to the website as a record of your sampling observations.
The next thing you want to do is make sure you've taken all of your site photos. So other than the photo of your data sheet, make sure you have your 2 site photos and your pocket microscope photo. And then make sure you rinse out your gear, rinse out and dry, and freeze your water sample bottle until it's transferred to project staff or sent back to UMD for further analysis.
Perfect. And finally, thank you for helping us keep a lookout on our lakes. Minnesota Lake Lookout on 3. 1, 2, 3.
Minnesota Lake Lookout!